There we go. Okay, so we saw that slide, went over kind of how to do, oh, I have a worksheet for the big one, the bigger dihybrid. Well, it's not a dihybrid cross, but it's kind of similar. Two characteristics to look at, yeah. I'll hand that out again. All right. So first I want to talk about a test cross, and this we already sort of looked at, or we definitely looked at when we did the, back to the front so you can hear me. We basically did a test cross when we did that other Punnett square worksheet and looked at the albinism. So when the child was albino, the mother was albino, and we had to figure out what the father was, if he was, what allele he had, basically, the other allele. So a test cross is an easy way to figure out what the genotype of one of the parents is. So you obviously know the phenotypes. If we're looking at flower color, for example, we have a purple flower and a white flower. If we know white's recessive, so you have to know about the system a bit. If we know white's recessive, we know this flower must be little p, little p. If we have a purple flower, we know that one has to have at least one big P. We just don't know if it has another big P or a little p. So if you do a test cross, you can look at the outcome and basically determine pretty simply what the genotype of the unknown genotype is of the parent. So that's the whole purpose of a test cross. You're just testing to see what the offspring ratio is going to be. If this parent had only big P's, you wouldn't get any white offspring, right? Because you don't have any possibility of getting two small p's, because the parent is always donating, the purple parent is always donating a big P. So you'll have all heterozygotes, so they'll all be purple. If you end up with this ratio, basically half and half, you know that the parent plant, or the purple one, sorry, must have donated a small p in addition to the big P that you know it has. So that's pretty logical and rational. Hopefully that makes sense to everyone. You can just work backwards, just like we did with that albino example. So yeah, the way to do this, you need a homozygous recessive individual. So you have to have an individual that can only donate the recessive allele, which would be a homozygous recessive individual. Cross them with the unknown parent, and then you look at the offspring ratios. And that's all there is about a test cross, really. It's an easy way to work backwards. So now we're going to get into the idea that inheritance is very complex. And we're not even going to get into all of the complexities. I'm going to go over some of the big different systems that inheritance, the different patterns that inheritance can follow. There's a lot of other exceptions as well. Dominance, basically, we're going to talk about situations where dominance doesn't mean a complete masking of the recessive trait. So that purple example, the dominant allele, the big P, wouldn't necessarily completely mask the little p. Both of them might show up a little bit. That's what we're going to get into here. So yeah, patterns of inheritance can often interact in an expression of a trait. So we'll talk about these two different ideas. I'll give you examples of these different systems, the names, and sort of how they work in general, and how you can tell that it is this specific system that we're talking about. So what we've been talking about is complete dominance. Just the simplest version, it's called complete dominance. Now we're going to do other stuff. So in total, there are 3 types of dominance. We have, what we were just talking about, complete dominance. That's what Mendel did. That's what he was looking at. I don't know if he got into these other kinds of dominance, but most of what he's known for is that idea of complete dominance. If there's a dominant allele, you don't see the recessive phenotype at all. That is not always the case. You can have these other 2 type of dominance systems called incomplete dominance and codominance. And they sound very similar, unfortunately. but they are slightly different. This image is a really great visualization of what incomplete, complete, and codominance are. So if you have your typical complete dominance scenario like we've been talking about, and we have, we'll say, a red allele and a white allele, the red allele being dominant to the white. You have a dominant version of the phenotype, red. If there's any dominant allele present, it turns out, the flower turns out red. If it's only the recessive allele present, it turns out white. Those are the only two options. It's red or it's white. With codominance, we'll start down here, you get a little bit of both. So you get some white and some red on the flower petals. So red and white both show up. So both of the alleles are seen as they would be in these two separate ones, but on the same flower. That's codominance. So co you can think of as equally dominant. Incomplete dominance is where you get a mixture. So red and white together makes pink. It's like blending paint colors. So incomplete dominance would be the blending of the two different phenotypes. Codominance, both of them show up as they would originally, yes.
You know how like for the red one it would be like if you said like uppercase F, uppercase F. Yeah. What would it be for like the bottom two?
I'll show you. Yeah, so it's a different, I'm glad you brought that up. It's different annotations. So we don't do the capital letter, little letter anymore. We have a different way to notate it because yeah, one's not dominant over the other. So that would be a little bit confusing. So yeah, we'll look at each of these now, the incomplete and codominance in a little bit more detail. So, okay, this is a complete dominance. We know what happens with complete dominance. Here we have, like Shay said, the big and little letters, uppercase, lowercase. We know kind of the pattern that happens. If something is heterozygous, it looks like the dominant phenotype. It has to be homozygous recessive to look like the recessive phenotype. Something else. There we go. So this is where these ratio numbers come into play. I mentioned the 3-to-1 phenotype ratio if you have a monohybrid cross. So that's where you have two heterozygotes, you're breeding them, and you're looking at the offspring. So there's always going to be 3 dominant phenotypes to 1 recessive. That 3-to-1 shows up in the complete dominance scenario. I think everyone's got that down. That's the most straightforward of them. Okay, incomplete dominance. So these other two systems. Now we're going to use superscripts or exponents, however you want to think of them, that actually are different letters superscripted on a common letter. So it looks something like this. So if we have, this goes for incomplete as well as codominance. It'll be the same way we notate the different alleles. So you have C, so we're talking about color. So C just stands for color here. You have, each one has the color characteristic, right? A gene for color. This one has the red and this one has the white allele. So for you guys on Zoom, here's the red allele as a superscript and then the white allele as a superscript. This keeps you from thinking that one is dominant over the other. They're actually completely equally dominant. That's how you can think of them. So now if you combine these two in incomplete dominance, so if you mix red with white, that's where you're like, in incomplete dominance, it's like mixing paint colors. So red with white, you get pink in the F1 generation. They're all going to be pink if you have homozygous dominant and homozygous red, homozygous white. And then you can move on from there. Here's the description of incomplete dominance. Move on from there and you can then cross two heterozygotes. So they're still called heterozygotes. They can be homozygous or heterozygous, but there's not really, not necessarily a dominant and recessive I should probably take those off of here. It's not really a dominant recessive situation. They're sort of equal. So here, if we look at this cross, we have two CR gametes coming together. So that's going to make a red flower. And then we have this combo, this heterozygous combo, CR and CW. We have two of them coming together. So we'll have two pinks. And then we'll have one with two white alleles. So homozygous for white. So now our phenotype ratio, it's not 3-to-1 anymore, like it was with the homozygous, the complete dominance situation. We have 1 red to 2 pink to 1 white. So that's going to be our 1-to-2-to-1 phenotype ratio. And that's when 2 heterozygotes are crossed. So for you guys on Zoom, here we have our red, 2 pinks. So that's the 2, and then 1 white. So 1-to-2-to-1. Okay. Does that make sense? Okay. Now, codominance, where you have both of those colors showing up in one flower. So camellias are pretty famous for this. If you know the camellia flower, they're pretty popular in horticulture. So you might have them in your yard or your parents' yard. So codominance both, if it's a heterozygote, if both of those alleles are present, both of them are expressed. It's not a mixture. It's just there's pieces of white and pieces of red. So we're not blending the colors. We're just interspersing them. I guess that's a way to think about it. And then these, it's the same idea. You get the same ratio for the same reasons as with the incomplete dominant situation. So 1-to-2-to-1 phenotype ratio. So this would basically be the heterozygote, right? And you have 2 heterozygotes if you cross 2 of the heterozygous parents, if you have a monohybrid cross. So those are the 3 different systems of dominance. That's really all I want you to know about them. Be able to, if I tell you kind of what the parents look like, and then I'll tell you what the offspring looks like, the phenotype, be able to tell me if that's codominance or incomplete dominance, and then also know the ratios. So if I say you have a 3-to-1 phenotype ratio based on these parents, you should know that's complete dominance. If it's 1-to-2-to-1, you know it's not complete dominance, basically. It's either incomplete or codominance. All right. So what does this tell us? Basically, the 3 types of dominance tells us that it's sort of complex, not shocking. Biology can be complex. Genes, alleles, and inheritance can be more complex than they appear. I spent a lot of time on the complete dominant situation because it at least gives you a baseline for understanding how the rest of it works. And then the phenotype ratio can tell us something about the parents. It can tell us about the character or the gene of interest. So you can work backwards, like I've talked about, and understand the inheritance pattern within a specific species just based on these different phenotype ratios. So that's why I was talking a lot about phenotype ratios. So phenotype ratios are important to remember, and then just it's a complex world out there. And it's more complex than just those 3 types of dominance, obviously. I'm going to get into a few other aspects of complexity here that relate a lot more to humans than flowers. Oh yeah. So yeah, I just wanted to make sure you guys know the expected or predicted outcomes so you can analyze a specific scenario. So like I said, you can work backwards. If you know you have a 3-to-1 ratio, phenotype ratio, you know you have complete dominance, etc. So just those ratios that I pointed out that I circled and I said, these are important, know those. All right, so a few more interesting complexities. So maybe you don't think incomplete and codominance is all that interesting. We'll move on from there. So I talked about all of these are basically deviations from Mendel. So Mendel did, the straightforward complete dominance situation. He laid the groundwork for all the rest of this. All of these deviate from what he would have predicted based on what he knew. So incomplete and codominance we just talked about. I'm going to talk about these next four fairly quickly over the next few slides, but multiple alleles, polygenic inheritance, pleiotropy, and then just the influence of the environment that can affect genetics as well. So I'll talk about how each of these create even more complexity in our world. Okay, first off, multiple alleles. Multiple alleles is basically there's more than two. So we've been talking about purple and white, or purple and white, tall and short, brown and blue. There's only been two options, right? You probably have an idea that there are sometimes more alleles than just two for a given gene. Our blood types are a great example of this. There's actually 3 different alleles that you can have in your blood type. So I'm going to sort of describe how blood type works. It's also an example of codominance, and you'll probably see why as I go through this. So I don't know how much you guys know about blood type, but you either have A blood type, B blood type, AB, or O, right? And then you have the positives and negatives, which is something totally separate. How many people know their blood type? Not many. Okay. It's a good idea to know your blood type. If you donate blood ever, they'll tell you your blood type. So I have type A blood. So that means my red blood cells have an A protein on the outside. So all of your blood cells, unless you're type O, have these little proteins studding the outside of them. It's just basically recognition proteins. So that's why you need a specific blood type. If you get a transfusion or you need blood put into you, you can't have some other blood type put into you again, unless you're a very specific type. You have to have the same blood type as you have, more or less, because these recognition proteins are basically going to lead to your immune system fighting off the invading blood if it's not the same kind. So these little purple things are going to be our A proteins. If you have B type blood, you have these different proteins, B type proteins on the outside of your blood cells. You can have both of them. So if you're type AB, you have the A protein and the B protein on the outside of your blood cells. If you're O, you don't have any. You have no proteins studding the outside of your blood cells. So those are the 4 different options basically. So you have A, B, and O, which is essentially no protein. So those are our 3 alleles. And if we look at a Punnett square here, so this is just one character, right? But we have 9 squares instead of 4. So it's a little bit different. So you can have the A allele, the B allele, or the basically no protein allele, the O allele. And then depending on which of those, so say your mother would be AB, and then your father would be AB as well. So if they can each donate an A and a B, these are all the different possible blood types that the offspring of the parents could have. So if you have an A and a B, you have AB type. If you have 2 A's, you're type A blood, etc. If you don't have any, type O blood is pretty rare because it's I guess it would be considered sort of that homozygous recessive. Type O is the universal donor. If you guys have heard of universal receivers and universal donors, so type O is universal donor. It doesn't have any proteins on the outside, so it doesn't set off a warning when it's put into someone who has another blood type. So me, I have type A blood, but I could have type O blood transfused into me and I'd be fine because my immune system is not going to recognize, there's no proteins for it to recognize to fight it off. Unfortunately, that means for people who have type O blood, they can only accept type O blood. They cannot accept any other kind because all of these other blood cells have those proteins which will kind of set off a warning in their system and they'll reject those blood cells. So the universal receiver, acceptor, I don't know what I guess universal receiver is going to be AB. And hopefully you can tell why that is. So if you have type AB blood, you can accept type A blood because you have those proteins. You can accept type B blood because you have those proteins. And like everyone, you can accept type O blood because there's no proteins. So it's sort of a complex system. Knowing your blood type can be helpful in case you're in some kind of major accident. In general, if they don't know your blood type, they'll give you type O. So if you have type O blood, if you find out you have type O, you should definitely try to donate more often. My husband has type O blood and he donates as much as he can because they always need type O blood in case they don't know what type blood the person in the accident or ER has. All right, so that's a long explanation for blood types, but blood types are kind of interesting. That's the multiple allele. We have 3 different options essentially of alleles there. Polygenic or polygenic inheritance is where more than one gene influences a trait. So polygene means many genes. Many genes are influencing one specific phenotypic trait. So height's a good example. I don't have an image of it here. Skin pigmentation is also another good example. Eye color falls into that category. I've talked about eye color as in blue and brown, like one and the other, but it can be, it's actually influenced by multiple genes. So what it comes down to is how many dominant alleles there are across these different genes. So here, if we look at skin pigmentation, so we'll imagine all of these are basically different genes. So the more dominant genes you have across these many, many genes, the darker your skin will be. If you don't have many dominant genes, you have a lot of recessive genes across whatever genes are playing into the skin pigmentation, the lighter your skin will be. Same for height. Obviously, it's not a discontinuous distribution of height. You're not either short or tall. There's everything in between. That's because there's a lot of genes that actually influence your height. So the hairline here would not be polygenic. You either have a widow's peak or you don't. So that would be the opposite. I just have that up there, or it's in that figure to show you what the non-polygenic would show. So widow's peak is inheritable. If you have a widow's peak, one of your parents probably has a widow's peak. Okay. Pleiotropy. Pleiotropy is sort of the flip of polygenic. So we were talking about many genes influencing one characteristic. This is where a single gene influences a lot of different characteristics. So it's just one gene, but it can have an effect across many different parts of the body. So Marfan syndrome, I don't know if anyone's heard of this. It's not all that common, but it essentially makes people really tall and lanky. And that's not to say everyone who's tall and lanky has Marfan syndrome. But if you have Marfan syndrome, it's essentially one gene that's affecting the connective tissue in your body, which is sort of connecting all your organs, your muscles. There's lots of different connective tissues throughout your body. So it sort of makes for looser connective tissue. So people are typically kind of taller and lankier. It can affect all of these different factors in a person, just one gene. So one gene can have all of these effects, very specific kind of phenotypic relationships or phenotypic effects. So issues with the eyes, teeth might be crowded, a high arched neck, something to do with the heart cavity, really long arms, long fingers, long legs. And then there's a lot of other risks that go along with it. So heart and blood vessels have a lot of connective tissue in them. So a lot of times people with Marfan syndrome have circulatory issues or heart issues, scoliosis, flexible joints. I guess that's not a bad thing, but most of these are kind of, can cause some longer term issues. There's a couple famous, well, I know of one, I don't remember her name, but a kind of famous female basketball player who had Marfan syndrome. Okay, So that's pleiotropy. So polygenic inheritance and pleiotropy, they're kind of the inversions of each other. That's how you can think of it. One gene versus many genes, one phenotypic trait versus many phenotypic traits affected. Okay, and then this is really cool. I find this interesting, but it's because I like ecological type stuff. So the environment can actually influence the expression of genes. So it doesn't affect what genes are there, but it can affect how those genes are expressed. So temperature often affects this, pH, a number of other kind of environmental factors can have an effect. A really great example is not only in turtles, but other reptiles as well. What temperature their embryo develops at can determine whether they're male or female. So if you think about sea turtles, everyone loves sea turtles, right? They lay their eggs in the sand and they're kind of piled on top of each other. So the eggs at the bottom are going to be probably a little bit more insulated and warmer. They will, gosh, what is it? I think they're more likely to be female than the eggs on top, because the eggs on top are a little bit colder. They'll actually turn out male. So it's not predetermined what the sex is going to be. Within this kind of 28 to 52 day period, that's when the temperature is going to affect what sex the turtle actually is. So that's kind of cool.
So you can trick so if you take one of these eggs, you can make it more.
Yeah, you can incubate it at a different temperature and then change the gender. Yeah, it's pretty cool. I don't know if it's for all turtles. I'm pretty sure, like I said, it's other reptiles as well. It's not just turtles. So anyway, I think that's cool. All right. Into sex-linked traits. I think I have 2 more slides. We'll just finish a little early today. We're just gonna do sex-linked traits. So focus for just a few more minutes. I know it's warm in here. So sex-linked traits are specifically associated with sex chromosomes, and we haven't talked about all the chromosomes. Everyone has, maybe we have, you have 23 chromosomes, 23 chromosome pairs. So you can basically lay them out. So 46 chromosomes total, 23 from your mom, 23 from your dad. So they figured out, essentially, they've mostly mapped the entire genome of the human species, and they know exactly what genes are on each of these chromosomes. So you can create what's called a karyotype, which I have down here. So this is a karyotype. It's a visualization of all of an individual's chromosomes. So chromosome 1 has specific genes on it. Chromosome 2 has other genes, and these are homologous, right? So one's from the mom, one's from the dad. There's 23 pairs. 22 of those pairs are autosomes. So 1 through 22. Those are called autosomes. Most of our genes are found on these autosomes. 1 pair are the sex chromosomes. This is what determines your gender at birth. So an X and a Y. So if you have 2 X's, you're a female at birth. If you have X and Y, you're a male. And there's very specific genes on these chromosomes. So I'm going to talk about some linkage issues that can pop up on these sex chromosomes in particular. So I'm ignoring all of these others. We're not going to talk about the rest of the autosomes. Okay, so yeah, like I said, at birth, if you have 2 X chromosomes, we consider that individual female, XY male. There's some deviations from this, which I think I'll talk about in the next lecture. So yeah, the father basically determines the biological sex of the offspring. Why is that? Yeah, exactly. So he is the only one who can donate a Y. The mother, no matter what, is donating an X. Women don't have anything else to donate. We can only donate X's. The father can donate an X or a Y. So if he donates an X, that's going to create a daughter. If he donates a Y, it creates a son. So chances are about 50-50, although I think there's slightly more females, at least in the US, than there are males. I don't know what that's about, but anyway, it's not a perfect 50-50 split, but statistically it should be. A little bit more about the Y chromosome. The Y chromosome is tiny. So this is the X chromosome. It's kind of the same size as all of the other autosomes, all the other chromosomes, which are autosomes. The Y is this tiny little chromosome that basically just has features that phenotypic characteristics that males typically express. Certain genes are only found on the X chromosome. So there's a lot more genes on the X chromosome. Some of them are not found on the Y chromosome, and some of them are not found on any other chromosome. So they're only on the X chromosome. So a lot of, well, several recessive diseases are found on the X chromosome. So hemophilia, have you guys heard of hemophilia? When, like, if you get a cut or a bruise, your blood doesn't clot. It can be really, really dangerous. So hemophilia is recessive, and that gene is found on the X chromosome. Color blindness, same thing. So these are often, color blindness and hemophilia are both a lot more common in males than in females. They're both recessive. So does anyone have an idea as to why they're more common in males than females?
You said color blindness?
Mm-hmm. Or hemophilia, yeah, both of them. Based on the genetics. So if you think about it, yeah, you're on the right track. It's recessive. So if it's recessive, let's say this is a male, right? Because we have an X and a Y. So if his mother donates that recessive allele for color blindness on her X chromosome, it's not on the Y chromosome.
Oh, that's why, okay, because
Right.
it's not equal if you have both X's or like cancel out.
Exactly. So if the mom donates the recessive allele, that son is going to show that trait. There's nothing else to mask it because it's not on the Y chromosome. So if this was a female and there were two X chromosomes, maybe the mom donated that recessive allele for color blindness, but the dad didn't. So now the daughter would be a carrier for it. She'd have that allele, but she wouldn't express the color blindness. So with two X chromosomes, you can mask that recessive allele. With only one X chromosome as a male, that can't be masked. So that's why those recessive disorders show up a lot more in males if it's on the X chromosome. So an X-linked trait or sex-linked trait, kind of used interchangeably, those show up a lot more frequently in males than females. That's the take-home message, and you can kind of figure that out from here. So if we have, this would be the dad, right, the X and the Y. The mom is donating the two recessive alleles. If they have a daughter, there's no likelihood of her actually getting or expressing that recessive trait because she has a big B from her dad. That is not the case down here because the Y doesn't have that allele. So the Y is kind of just, you can just ignore the Y. The son is no matter what going to get that recessive allele because his mom has two X chromosomes with their recessive alleles. Make sense? Okay. Yeah, let's just stop there today. I think that's good. We'll pick up, no class Friday. So enjoy your long weekend and I'll see you guys on Monday.